Hi, all the 2021 SPM candidates from Cebu. Welcome to program Kolaborasi Kecemerlangan SPM 2021 organized by PPD Cebu and Persatuan Pengetuan Sekolah Menengah Cebu. I am Cikgu Sun Shuqing from SMK Bukit Asih, together with Cikgu Ling Biyan from SMK Kampung Nangka. We'll do some sharing about KSSM Biology Paper 3 to all of you. KSSM Biology Paper 3 tips sharing session consists of 3 slots. Slot number 1, the main focus is KSSM Biology Paper 3 format. Slot number 2 is about science process skills. And slot number 3 is about the manipulative skills and the precaution steps. All the sharing in slot number 1 and 2 are based on the information given in book of Format Pentafsiran Biology from Lembaga Peperiksaan. And the answering techniques are based on the previous Biology Paper 3. Slot number 1. Now we have a look on the KSSM Biology Paper 3 format first. Actually, the practical test is integrated. What does integrated practical test mean? In BM, we say bersepadu, meaning that during the practical test, each candidate will be given 3 items, which are biology, physics, and chemistry in one go. Candidates will answer the question one by one based on the subject they are registered. It can be 3 pure science subjects, bio, physics, and chemistry, or other combination of any 2 pure science subjects. There are 15 marks per item and 45 minutes per item. For the first 5 minutes, it is for experiment planning, and 40 minutes for conducting the experiment and answering the questions given. I have quoted some important information from Format Pentafsiran Biology book. Now we have a look. Okay, number 1. Calon boleh memulakan eksperimen daripada mana-mana soalan. Sekiranya calon tidak dapat meneruskan eksperimen selepas 15 minit, calon boleh meneruskan eksperimen di soalan yang lain. Jika calon masih tidak dapat meneruskan eksperimen di setiap bahagian, calon bolehlah berjumpa ketua pengawas berkecuali dan meminta pertolongan. Sebarang bantuan yang diberi akan dilaporkan dan sedikit maka mungkin dipotong. So from here, we can say that all the candidates are given chances to try their best in the practical test. So please do seek help if you really cannot continue the experiment in each item. Don't give up. Now we go to slot number 2, which is about the science process skills that might be tested in Biology Paper 3. Overall, there are 12 science process skills listed here. Number 1, observing. Number 2, classifying. Number 3, measuring and using number. Number 4, inferring. Number 5, predicting. Number 6, communicating. Number 7, using space-time relations. Number 8, interpreting data. Number 9, making operational definition. Number 10, controlling variables. Number 11, forming hypotheses and experimenting. Okay, and there is one more, which is about the precaution steps. So these are the science process skills listed in Format Pentafsiran Biology book. number 1 until number 12. Some of the skills will be tested in biology item, and some will be tested either in physics or chemistry. Among the 12 science process skills listed here, which one is the very first that we have to know or to master first before we start experimenting? The answer is identifying and controlling the variables. Here is the sample question given in Format Potassium Biology book. Now, let's look at the information given here. You have to carry out an experiment to study the effect of different concentration of solution on spinach stem. Spinach stem cutting should be cut longitudinally as in diagram 3.1. Are you able to find out the MV, manipulative variable, RV, responding variable, and CV, constant variable, from the information given here? Please try. Okay, so the MV is the concentration of solution, and the RV is the final look of the spinach stem, whereas the CV is the initial length of the spinach stem. Now, we look at the question given. Plan your experiment by using the apparatus and materials provided. Your procedure should include method to handle variables, precaution steps. So, any ideas how to write the method to handle the variables and the precaution steps? Good if you have the ideas. If not, please stay tuned. Now we go to question B1 and B2. B1. Carry out the experiment. State your observation on the spinach stem cutting in solution Z. B2. Explain your observation in B1. Here, we are tested about the skill in making observation and also making the inference. Question C1. Draw your observations in the space provided. From here, we can see that observation can be in a sentence and also can be in a form of drawings. Okay, now we go to question C2. Classify solution X and Y into their types of solution. So the science process skills tested here is classifying. Question D. The information below shows method to use vegetable fertilizer. Now we look at the info given in the label. Baja organic fertilizer. Usage instruction. Dissolve 5 ml of fertilizer into 500 ml of water. A housewife mix 20 ml of fertilizer solution with 500 ml of water to fertilize her vegetables. Predict what will happen to the vegetables after 2 days. Prediction, explanation. So the question D, science process skill tested here is prediction. And after we do the prediction, we have to explain why our prediction is like this or like that. Now, please look at this example. An experiment was carried out to determine the effect of different concentration of sucrose solution to the length of the potato strip. Figure A shows the setup of apparatus for this experiment. By using a cork borer, a cylindrical potato strip was obtained. The potato strip was cut at 5 cm long. So please observe the diagram A. Still remember the first must-do job in answering biology paper 3? Yes, look for the variables. So now, please write down the variables based on the information given here. Please write down so that we can use it for discussion later on. Some more information about the experiment. Please read the information given carefully. The final length of potato strip was immersed in 0.1 m sucrose solution after 30 minutes is shown in figure B. The experiment in figure A was repeated by using different concentrations of sucrose solution. Able to write the answers for the variables, which are the MV, RV, and CV? Please try. Now we look at the answers for the variables. Number 1, the MV is concentration of sucrose solution. The RV is final length of the potato strip. The CV is initial length of the potato strip. Able to get all correct? Congratulations if you have tried your best to answer it. Now we discuss the method to handle variables. Please look at the highlighted keywords in the suggested answers. Number 1. For the MV, use different concentration of sucrose solution. Number 2, the RV method to handle is measure and record the final length of the potato strip by using meter rule. Number 3, the CV method to handle is fix the initial length of the potato strip at 5 cm. In order to write the method to handle variables correctly and easily, we can always follow the tips given here. For the MV, the verb use, so we write use different MV. For the RV, the verb measure and record. The record is compulsory in the answer. So measure and record the ORV, the operating responding variable, means the RV that we can observe in the experiment by using an apparatus. We also can write calculate the RV by using formula. When we are using the verb calculate, we must include the formula in the answer. For the CV, the verb such as fix or use the same in the method to handle together with the particular value. Now, we look at the hypothesis. Hypothesis is a statement that shows the relationship between the MV and the RV. Based on the information given just now, here is the hypothesis that we can make. When the concentration of sucrose solution increases, the final length of cylindrical potato strip decreases. So the first part is about the MV, and the second part is about the RV. And the relationship is increases, decreases. So the formula to write hypothesis correctly is MV plus RV plus the relationship. There are two ways of writing hypothesis. Number one, we can write in this way. MV increases, RV increases. Number two, MV1 has higher RV than MV2. For number one, the MV is the variables with values such as temperature, time, length. For number two, the MV is without value. For example, the food samples. So if we have cashew nut and the dry bread, we want to compare the energy value between these two samples, then the hypothesis is cashew nut has higher energy value than dry bread. Now we look at the final length of the potato strips in different concentration of sucrose solution. So here we have the 0.1 m, 0.2 m, and the reading is 5.9 cm, 5.6 cm. Please do write down the first reading now, which is 0.1 m, and the final length is 5.9 cm. Some more data now, which is 0.3 m, 0.4 m, 0.5 m, and the reading 5.3, 4.6, and 4.2. Please do write down the last reading now, which is 0.5 m and 4.2 cm. Based on the data that you have recorded just now, which is 0.1 m, 5.9 cm, and 0.5 m, 4.2 cm, please try the observation. Please do have a try. Can do it. Now we look at the answer. When the concentration of sucrose solution is 0.1 m, the final length of potato strip is 5.9 cm. Do you have similar answer as mine? Now we look at the second answer. When the concentration of sucrose solution is 0.5 m, the final length of potato strip is 4.2 cm. Can get the idea how to write the observation? Here, two science process skills they are using now, which is measuring and using number, and also making observation. And the correct technique in writing observation is we should include the MV, the manipulative variable, and the operating responding variable together with the values. We look at the example as shown as above, which is the concentration of sucrose solution is the MV, and the final length of the potato strip is the operating responding variable. And we include the values, which is 0.1 m and 5.9 cm. With this, our observation is a complete observation. And another observation can be drawing with label. In a quantitative observation, we have to include the MV, RV, and the value as shown in the first formula. Whereas in a qualitative observation, we might be asked to draw a label diagram as the observation. Everyone so far can follow? So for this part, should be not a big problem for most of you. Am I right? After observation, candidates mostly will be asked about the inference or explanation. Means we need to give reasons why the observation is like this or like that. Now we look at the inference correspond to the two observations that we have made just now. Number 1, 0.1 m sucrose solution is hypertonic to the cell shape of the potato. Therefore, water molecules diffuse into the vacuole of the potato cells. Number 2, 0.5 m sucrose solution is hypertonic to the cell shape of the potato. Therefore, water molecules diffuse out from the vacuole of the potato cells. The first inference is corresponds to the first observation. The first inference will try to explain why the final length of the potato strip is much longer than the initial length by relating to the 0.1 m sucrose solution is hypertonic to the cell shape. That causes the water molecules diffuse into the vacuole. The second inference is correspond to the second observation. It tries to explain why the final length of the potato strip is much shorter than the initial length by relating to the 0.5 m sucrose solution is hypertonic to the cell shape. Then the water molecules diffuse out from the vacuole. Now we go to communicating. Means we will present the experiment data in the form of table or graph. This is the instruction for constructing a table. Construct a table and record all the data collected in this experiment. The table should have the following titles: the concentration of sucrose solution, the initial length of potato strip, the final length of potato strip, the percentage changes in the length of potato strip. This is the suggested answer. We only need to rewrite the titles given in the table. Column number 1, the concentration of sucrose solution with the unit m. Column number 2, the initial length of potato strip and the unit is cm. Column number 3, the final length of potato strip, also cm. And the last column, the percentage changes in the length of potato strip, percentage. So unit must be included in the title. Then the data is recorded accordingly. So the data 0.1 m, the initial length 5 cm, the final length 5.9 cm. All these recorded accordingly. And the last column, we need some simple calculation to get the answer. So for the first reading, the percentage change in the length of potato strip is 18%. And for the last one, 0.5 m, the percentage change in the length of the potato strip is negative 16%. Once the table is completed, then we can draw a graph of y against x. This is the instruction. Use the graph paper, draw the graph of the percentage change in length of the potato strip against the concentration of the sucrose solution. At the x-axis, we must label with MV and unit, which is concentration of the sucrose solution, M. At the y-axis, we must label with RV and unit as well, which is the percentage change in the length, %. After drawing the graph, then we can state the relationship between the MV and the RV based on the graph plotted. Now, please look at the question. Based on the graph, state the relationship between the percentage change in length of the potato strip and concentration of the sucrose solution from 0.1 m to 0.3 m. So how do we answer this question? Now we look at the suggested answer. When the concentration of the sucrose solution increases from 0.1 m to 0.3 m, the percentage change in length of the potato strip decreases. Always remember, we must write the MV, then followed by the RV. Now we go to interpreting data. The question can be like this. Based on the graph, state the concentration of the sucrose solution, which is isotonic to the concentration of the cell set of the potatoes. Explain. So how do we answer this question? We must refer to the graph. And the answer is the concentration of sucrose solution, which is isotonic to the concentration of the cell set of the potato is. So the answer must refer to the graph. We must find out the point that the graph passed through the x-axis. So we get the point from there as the answer. And we have to explain also the answer. So this is the suggested answer because at the particular concentration, there is no changes in the final length of the potato strip. Water molecules diffuse in and out of the potato strip at the same rate. So this is the proof of the isotonic. The solution is isotonic to the potato cell set. Is everyone so far so good? Which one is your condition now? Hopefully everyone is still very energetic. Now we discuss about the operational definition. Based on this experiment, state the operational definition for the osmosis process. So how to answer the question? Now we look at the suggested answer. Osmosis is a process when the potato strip immersed in sucrose solution, which cause water molecules diffuse in or out of the potato strip across the plasma membrane. Osmosis is shown by the final length of the potato strip. Osmosis is affected by the different concentration of sucrose solution. Please pay attention to the answering technique in the operational definition by focusing on the highlighted wordings. So tip number 1, we have to put in the materials that being used in the experiment. For example, potato strip, sucrose solution. Tip number 2, we have to put in the RV that being observed by writing shown by. So the ORV is the RV that we can observe. O here is the operating responding variable. And tip number 3, we have to put in the MV by writing affected by. So the MV in this question is the sucrose concentration of sucrose solution. Therefore, we write like this: osmosis is affected by the different concentration of sucrose solution. Now we go to predicting. The question is, if the experiment is repeated by using 0.8 m sucrose solution, predict the final length of the potato strip. Explain. In the previous experiment, the range of the concentration of the sucrose solution is 0.1 m to 0.5 m. Now, the concentration of the sucrose solution in the repeated experiment is 0.8 m, which is much higher than 0.5 m. So, what is your prediction? The final length of the potato strip increases or decreases? Please predict. Yes, the final length of the potato strip decreases. So we can write it this way. The final length of the potato strip is 3.9 cm. And we have to give reason. We have to explain. Because 0.8 m sucrose solution is hypertonic to both potato cell sap and also 0.5 m sucrose solution. More water molecules will diffuse out from the potato cell sap as compared to the potato strip that immersed in 0.5 m sucrose solution. Therefore, the final length of the potato strip is shorter than the potato strip in the 0.5 m sucrose solution. Now, we go to the last science process skill, which is classifying. Here is the question. In another experiment, the mustard stem strip were immersed in different concentration of sucrose solution, which are 0.2 m, 0.4 m, 0.6 m, and 0.8 m. This is the condition of the mustard green strip after immersed in different concentration of sucrose solution for 30 minutes. Please study the diagram carefully. Please look at the shape. In the table below, classify the final condition of the mustard green strip based on the different concentration of sucrose solution that they being immersed. Everyone, please have a try based on the small diagram shown at the left hand side. So we have to classify according to the concentration of sucrose solution. Please exercise your brain. Okay. Already have the answers. Now we go to check the answer. Can get all correct? I believe many of you can answer well. Am I right? Before we proceed to slot number 3, there is a self-test quiz for all of you. This is to test how good you are in the science process skills. This is a question in the SPM 2009. Now, please study the information given. We read together. A group of students carry out an experiment to study the effect of light intensity on the rate of photosynthesis. Diagram 1 shows the apparatus set up to collect the gas produced when the aquatic plants receive different intensities of light. The aquatic plant was illuminated from a light bulb of 40 watts for a period of 5 minutes at 5 different distances d from the bulb. At the end of each 5-minute period, a gas bubble was drawn into the capillary tube using a syringe. The length of the gas bubbles was measured using a ruler and recorded. The temperature in the water bath was maintained at 25 degrees Celsius throughout this experiment. So everyone, please try to do the first job in the question. What should we do is, can you remember? Yeah, we have to identify the variables. So now, study carefully. Try to find out the MV, the RV, and the CV from here. Please have a try. Now, please compare your answers with this. Number 1, we look at the MV. The MV is the light intensity. We also can say the MV as the distances from the aquatic plant and the light bulb. Number 2, the RV is the rate of photosynthesis. Or we can also say that the length of the gas bubble. Number 3, the CV. There are several answers here. Number 1, power of the bulbs, which is 40 watts. Number 2, the illumination duration, which is 5 minutes. And number 3 is the temperature in the water bath, which is 25 degrees Celsius. Are you able to identify all of the variables listed here? The diagram here shows the apparatus setup in the experiment. Please study the diagram and try to recall what we have discussed just now. From this diagram, we can identify another CV, which is the concentration of sodium hydrogen carbonate solution. Here is the data collected. distance from the bulb and the length of the gas bubble. So the distance from 50 cm decreased into 10 cm, whereas the length of the gas bubble from 1 cm increased to 5 cm. Now we will try to write the observation based on the data given. Still remember how to write the observation? Please have a try. The formula is MV, RV, and values. Done? Now we look at the answers. We can write in this way. When the distance of the bulb is 50 cm, the length of gas bubble is 1 cm. The second observation, when the distance of the bulb is 10 cm, the length of gas bubble is 5 cm. So always remember the observation is MV, RV, and the value with the unit. Now, state the inference. How to write the inference? Can all of you still remember? This is the way. At a distance 50 cm, the light intensity is low, the rate of photosynthesis is low. And the inference for the second observation is light intensity is high, rate of photosynthesis is high. So here we want to explain why the length of the bubble is like this or like that. And the formula for the inference, MV, RV plus the description. Hopefully everyone can remember this. Now we go to method to handle the variable. Based on the sentences given here, are you able to fill in the keywords for each method to handle variable? Please have a try. Here is the answers. For the MV, place or put the bulb at different distance, which are 50 cm and so on from the plant, or use different light intensity. For the RV, the method to handle, measure and record the length of gas bubble by using a ruler, or calculate the rate of photosynthesis by using formula, which is the length of bubbles divided by time. For the CV, we fix the temperature at 25 degrees Celsius. Now everyone please try to state the hypothesis based on the information given. Please have a try. We have the MV, the RV, and the relationship in the hypothesis. Okay, now we look at the answer. When the light intensity increases, the rate of photosynthesis increases. Or, when the distance of the bulb increases, the length of gas bubble increases. Are you able to do so? For construct a table, normally we are given the titles and we just construct a table based on the titles given, then put in the data. So to save time, I just give you the table with the data and also the calculation of the rate of photosynthesis. So the unit still compulsory in the table. Based on the data, now draw the graph of rate of photosynthesis against the distance from the bulb. Three main aspects that we need to be careful. Number one, we must use the uniform scale on both axes, and all points plotted must be correctly and able to join all points to form a smooth graph. So all the candidates, you may draw the graph after this session and let your teacher to check the graph for you. Once you have plotted the graph, now based on the graph, explain the relationship between the rate of photosynthesis and the distance from the bulb. So this is a homework to all of you also. Operational definition. State the operational definition for the process of photosynthesis. Please try this after this session also. Now, prediction. Another group of students carried out the same experiment. The aquatic plant was exposed to a similar lighter bulb at a distance of 30 cm. Using 0.05% sodium hydrogen carbonate solution. Predict the outcome of this experiment and explain your prediction. So this is another homework. Lastly, classification. Classify the following into groups of requirements to carry out the process of photosynthesis and the products obtained. Oxygen, carbon dioxide, chlorophyll, light, water, glucose. Classify it according to the requirements and products. Please try after the session also. Now let's recall the 12 science process skills that we have discussed before we move to next lot. So here there are a total of 12 science process skills: observing, classifying, measuring and using number, inferring, predicting, communicating, using space-time relations, interpreting data, making operational definition, controlling variables, forming hypotheses, and experimenting. So the experimenting means the conducting of the experiment. And during the experimenting, there are some precaution steps that we have to bear in mind. So this will be discussed in the next slot. Now we go to slot number 3, which is about manipulative skills and precaution steps.